Okay, so I'm going to talk today in regard to XXY as one of the conditions that we classify in X and Y chromosome variations. So to start, I wanted to talk a little bit about terminology that we use in these conditions in the healthcare field. When we talk about chromosomes, we're talking about the structures that carry our DNA, that encodes our genes and tells our bodies how to grow and how to develop. And when we use the term aneuploidy, we're talking about either an additional or a missing chromosome that can be present in the cell or all cells of the body. When we're talking about sex chromosome aneuploidies, abbreviated as SCA, we're talking specifically about having either an X or a Y chromosome that is additional or missing in the cells. And when we talk about sex chromosome trisomies, we're talking about the presence of 3 sex chromosomes, or those X's and Y's, having 3 copies of them. And the common conditions that we talk about with this are Klinefelter syndrome, also known as XXY, Jacob syndrome, also known as XYY, and trisomy X syndrome, also known as XXX. And this class of conditions really has a lot of different names, but for the most part, they all really refer to the same class of conditions. We're talking about sex chromosome aneuploidies, which is a very scientific term that you may encounter when you're reading about these conditions, but they can also be referred to as sex chromosome abnormalities or anomalies, disorders, or variations. And then some individuals talk about them as X and Y chromosome aneuploidies or X and Y chromosome variations, which I often hear from families is a term that they prefer, and it tends to be a little bit more accurate. In the class of X and Y variations, there's a lot of different types of variations that can be present. So while we're going to be talking today a lot about XXY, XXY is one of many different types of variations. And the larger circles represent the trisomy conditions that tend to be more common in our population, but there are other conditions in which there are multiple extra X's and Y's that can be present and that we see commonly in our clinic. Combined, these conditions are really common. So they can occur in about 1 in 400 live births, the most common of which is Klinefelter syndrome, being 1 in 650 males, followed by Jacob syndrome or XYY, as well as trisomy X or XXX that occur in 1 in 1,000 males and females respectively. And then the other types of sex chromosome aneuploidies that we'll commonly see are more rare and won't be really focused on in this talk today, but we do talk a lot about overlapping conditions and presentations and features that we can see in our clinic with these conditions as well. So to give a little background about Klinefelter syndrome, it was first described by Dr. Harry Klinefelter, who is an endocrinologist, in 1942. And Dr. Klinefelter published a case series of 9 adult men who presented with similar physical features, including testosterone deficiency. And it was identified in 1956 that Klinefelter syndrome was actually attributed to the presence of an extra X chromosome in the cells. So this photograph shows that I'm holding a portrait of Dr. Harry Klinefelter, which was gifted to our clinic several years ago as a huge honor. His daughter, after he passed away, had reached out to us and offered to donate his portrait to our clinic, which we very proudly hang on the wall of our office. What we know about Klinefelter syndrome not only is from Dr. Klinefelter's initial description, but we really learned a lot when newborn screening studies were initiated in the 1970s as well as the early 80s. Historically, a lot of what we knew about XXY was based on how it was identified, right? So there's a publication by Abramsky et al. That talks about how different indications of testing really identify different portions of the individuals with this condition. But the majority of individuals actually never are diagnosed in their lifetime, accounting for about 65% of individuals with XXY. And secondary to that, the majority of individuals who are identified during adulthood, during workups for infertility, in which they're identified as either having hypogonadism or small testicles, as well as inability to achieve a pregnancy. The other attributes or the other aspects that can be identified in the individuals historically included anything from prenatal testing to developmental delays to lack of pubertal progression. So it really is important to note that a lot of what we know historically about XXY is based on a very small percentage of the population in total that was identified. Today, we're seeing a big shift, though. So there's a new technology called non-invasive prenatal screening. And this was initiated in 2013, becoming commercially available. And it's a screening test. It's not a diagnostic test. But this screening test looks at fragments of fetal DNA that are shed from the baby's placenta during pregnancy and into the mother's blood. And this is commonly found as a percentage of all fragments of fetal DNA and circulating cell-free DNA about 10 weeks and later in pregnancy. So this is a test of the mother's blood. There's no risk to the pregnancy as far as causing a miscarriage, which is why it's referred to as a non-invasive prenatal screen. And when we're testing this, we're testing for these fetal fragments. It's a test that's also commonly referred to as cell-free DNA testing. And like I said, it became available in 2013, but as of 2020, the American College of Obstetrics and Gynecology recommended that all women, regardless of maternal age, be offered this type of screening test as it posed no risk of miscarriage to the developing fetus. We know that the best time to test the gestation or the pregnancy with this type of technology is at 10 weeks or later, and that's because fetal fraction or the concentration of cell-free that is from the fetus in the maternal blood is high enough to have a quality result. The results from this screening test, knowing that it's not diagnostic, can either be positive, where it identifies an increased risk for a condition, negative, in which it does not identify an increased risk for a condition, or inconclusive, in which they're not able to make a call from the results of the test from the laboratory. There's a lot of variability on the methodologies that labs will use in doing this test. So not all labs approach non-invasive prenatal screening the same. And in XXY as a screening test, it has a positive predictive value of about 74%. So positive predictive value is really important to understand as a screening test. Positive predictive value means if a test comes back as positive, what is the likelihood that it truly is a positive when diagnosed, right? So how likely is a positive screen going to result in a positive diagnosis? And in XXY, that's only about 74%. This is a statistic that's taken from a recent publication from the American College of Medical Genetics, looking at a meta-analysis of 28 different studies. And the numbers really ranged anywhere from 59 to 84% when they analyzed the studies from an XXY perspective. But this really highlights that the non-invasive prenatal screening is not a diagnostic test, and decisions about the pregnancy shouldn't be based on this result alone, that it really does need, at some point, either prenatally or postnatally, diagnostic testing to confirm if it's a true positive or a false positive. It's also important to remember that, and we've seen this clinically in a few cases, that discordance is possible. So what that means is that there could be an underlying condition that's different from the initial screening results, but still abnormal. So when we have a screening result that identifies an increased risk for XXY, how do we diagnose? How do we proceed? In the two genetic testing procedures that are followed after a screening result are really either prenatal or postnatal. So during pregnancy, an individual could proceed to having either a chorionic villus sampling, called a CVS, which is a sample of the placenta. And this can be performed anywhere around 10 to 13 weeks of gestation, or an amniocentesis that takes a sample of the amniotic fluid from around the baby And this is performed 15 weeks or later for diagnostic testing of a genetic sample from the fetus. The other option is deferring until postnatal. So after the baby is born and having testing either by a blood test or a cheek swab where you're collecting with a little brush from the inside of the cheek, or a sample of saliva that will contain cells from the individual and be able to do the analysis. And the analysis that's conducted either prenatally or postnatally is either a karyotype or a microarray. So a karyotype is a genetic test that looks at the count and structure of the chromosomes in cells. And so in this slide, you can see pictures of a single cell where the chromosomes are stained. And then they take the chromosomes and put them into an orderly picture called a karyotype that puts the chromosomes in their respective pairs. So an individual typically has pairs of chromosomes because they get one chromosome in each pair from each parent. So in the first pair, we have one from mom and one from dad, second pair from mom, from dad, and so forth and so on. And the chromosomes are numbered 1 through 22. When we're talking about our sex chromosomes, we're talking about this 23rd pair. And they're labeled either with an X or a Y. The Y chromosome carries the genes that drive male development. In the absence of the Y chromosome, we have 2 X's, and that drives female development. When we have an individual with Klinefelter syndrome, their karyotype demonstrates the presence of an extra X chromosome in this 23rd pair. And that's diagnostic of Klinefelter syndrome, or 47XXY. The other way that we can study chromosomes or diagnose XXY is from a chromosomal microarray. So this test obviously looks very different from what I was showing you before. And it uses probes that attach along the individual chromosomes, the first pair, the second pair, the third pair, and so forth. And it's balanced, representing 2 copies. So the straight red line representing 2 copies of the first, 2 copies of the second, 2 copies of the third. What we're focused on is actually having an overrepresentation when we're looking at either the X or the Y. In XXY, we would see an overrepresentation of the X chromosome. Having 2 X's, this is a little misleading because this is actually a diagnosis of trisomy X, but the idea is still the same in that you see a higher level of the X chromosome represented than you would anticipate in a typical XY male. So what causes XXY? Well, XXY results from a chromosomal non-disjunction. And so what does that mean? So if we have a cell in the body and we want to take one cell and have it divide into 2 cells that are equal to the initial cell, the chromosomes duplicate and then they have to divide into those 2 new cells. And so if there's any kind of failure of the chromosomes to separate evenly during that cell division process, that's called non-disjunction. And we can result in having either an extra or a missing chromosome in one of those daughter cells, if you will, or the next generation, those 2 cells. This can happen either in forming the egg or the sperm. And what we see with XXY is about half the time, it results from having an extra X chromosome in the egg, and the other half the time it results from having an extra X chromosome in the sperm. We can also see this non-disjunction occur after conception. So the sperm and the egg come together at conception. There's a normal number of chromosomes, and as those cells grow and divide, that if the chromosomes fail to separate properly during those cell divisions, we can end up with mosaicism, which is reflective of more than one result in an individual. So some cells have one finding, other cells have a different. But it's not caused by something that a parent would have done either before or during pregnancy. This is not your fault that the child has a diagnosis of XXY. There are many different types of chromosomal conditions in which non-disjunction can occur, and it's not the result of something that the parents did. There's a lot of variation that we can see within the condition of XXY. So it would be a whole different talk to just talk about these features, but at kind of a higher level, there's a lot of variation that we can see in physical features associated with XXY, as well as developmental features, medical features, and psychological features such as learning and behaviors. And when we look at some of the features in comparison to the other conditions, so we're talking today about XXY or Klinefelter syndrome, but in comparison to some of the other trisomy conditions that we can see, there's a lot of overlap that we can see from a neurodevelopmental perspective and the risks that can be associated. So we know that XXY has a risk for having developmental delays, learning disabilities, ADHD and executive functioning deficits, anxiety, social communication problems or autism, and motor skills disorders. We also see that there's more strengths when we look at the learning profile of individuals with XXY and more of the nonverbal and spatial skills, and we see more areas of weakness or learning disabilities more in language-based or verbal skills. When we talk about the medical features associated and the physical features associated with XXY, we know that individuals tend to be taller, a couple inches, about 2 to 3 inches taller than their what we call mean parental height, or what their expected height would be based on their parents' heights. We also know that there's testosterone deficiency due to testicular dysfunction, as well as infertility. And we know that birth defects tend to be rare in XXY and not higher than the general population. So the unique feature I alluded to in regard to testicular dysfunction that we can see not only in XXY, but really any man that has an extra X chromosome. And that can be one extra X, or that can be multiple extra X's. So the testicular dysfunction that we see is kind of in two categories. So we see that the testicles aren't able to make enough testosterone, and that usually universally occurs more in adolescence. So when we talk about testosterone in individuals, and we compare boys that are typical XY to boys with the extra X chromosome, what we see is a little bit of a difference at the two periods in which testosterone will naturally surge. So infants will have a testosterone surge around 3 to 6 months of age. And there's been very small studies on this level, but what we're seeing is even in these small studies, there's some risks of having a lower peak of the surge when we're looking at boys with XXY. Now, we still have a lot to learn about the role that this infancy testosterone plays. The body naturally going into childhood does not produce a lot of testosterone. And then testosterone starts to be produced associated with puberty. And all boys, even with XXY, will start puberty typically just fine on their own. But what we can see over time is that their level of testosterone tends not to rise to the level of a typical boy, indicating that they need testosterone replacement. Testosterone is a hormone that you'll hear about in other talks, but it is really important for overall, not only physical development, but long-term health, especially with bone density. The other aspect that we see that's unique when a man has an extra X chromosome is infertility. And this is a topic in which we describe and talk about it very specifically, that there is an absence of sperm in the semen of a man with an extra X chromosome the majority of the time. Now, there are some procedures that are offered and successful in about half the cases. If a man goes today to see a reproductive specialist and in the office and during the appointment says, I have XXY and I'd like to see what my options are, that today the procedures that are available to men are successful about half the time in achieving a biological pregnancy. And I anticipate that in the next 20 to 30 years, given the amount of research that is dedicated to this space, that we will see a lot of changes over time. So the perspectives on XXY is that this is a very highly variable condition. We can see a wide range of how our patients can present when they have XXY. And we also recognize that family background genes and the environment in which an individual is raised can really have a significant impact on outcomes. We know that when individuals are identified early, that the family is able to have a very proactive approach, whether it's accessing therapies or having evaluations around learning that can have a significant impact on long-term outcomes. And then just keeping in mind that children are individuals, and individuals have various strengths and weaknesses that are unique to who they are as a person. It's not all attributed to XXY. And we see a lot of strengths that are beautiful in many of the patients that we follow in clinic. So when we talk about genetic counseling, and I'm not going to dive too deep into this, I just want to have the perspective that genetic counselors can really play an important role with families for a variety of other aspects in addition to the education around this condition. So they can help families in planning next steps after diagnosis. They can help identify if symptoms are attributed to the diagnosis, or equally important, if they're not attributed to the diagnosis and additional evaluations are warranted. They can help talk about disclosure of the diagnosis, whether that's to the individual or to the other family members or to the school. Also helping advocate for services in school if a child does have learning disabilities and needs assistance. They can help in navigating healthcare as far as resources and getting care for the child, as well as resources in transitioning to adulthood that are very personalized in need, given the variable aspects of this diagnosis that we can see. And then connecting to community resources and a lot of the resources that are available to individuals and families. And then talking to individuals in adulthood about recurrence risks and having children. There's a variety of resources that are available in supporting families with XXY. AXYS has an amazing amount of resources available on their website. Not only this recorded webinar, but also other recorded webinars over the years that are available. There's a lot of publications that are available, including a book written by Virginia Cover that is a wonderful resource to families with XXY. Our clinic has a wonderful website as well, with published medical journal articles that we've made available, as well as other resources about sex chromosome aneuploidies. And then Amazon has some resources available that we've worked in developing through our clinic with our students, including a series of children's books about the conditions, including XXY. And we're hoping in the near future to have additional resources available through Amazon as well for new families. So with that, I'm happy to take any additional questions that anyone may have. And I thank you for your time.